dear registrants and dear all, it's really a great pleasure to have you today on our meeting and our HDN Roche platform meeting. And today we will discuss the medication called tominersen and the results, the recently published results of the clinical trial Generation HD1. We will also touch upon the currently active clinical trial Generation HD2. And I'm delighted to introduce our distinguished speaker, Peter McColgan, and also the panel members that will take part in the panel discussion. So the panel is chaired by Jean-Marc Burgunder, and the panel members are Sarah Tabrizi, Ed Wild, Anne Roser, Bernard Landwehrmeyer, Patrick White, Blair Levitt, and Frederic Sandu. So with this, I guess I will pass the word to Jean-Marc.
Oh, well, to me, that's great to be here to have the opportunity to discuss this data. And I will not make a long introduction because you have done it already. And we are now very interested to look at the new data and have this presentation by Peter. We do appreciate the way Roche is very open to have those type of discussions to set the stage for us. And Peter, the floor is yours. You can present your presentation, please.
Thanks so much, Jean-Marc. Give me a second. Let me just. So I'd just like to start by thanking the EHDN and the organizers and the panel for inviting me to be able to cover Generation HD1, but also talk about the path forward for tominersen and how what we've learned has really formed the basis of the current study. So for those that don't know you, my name is Peter McColgan, and I'm the clinical science leader at Roche on the tominersen program. Just begin with my disclosures. And I want to give an outline of today's presentation. So I'm going to give an overview of the tominersen program, then focus on the key findings from Generation HD1, and then delve into some of the post hoc analyses that help us understand what happened on the program. And then move to Generation HD2, which is our current phase 2 study that's well underway. And I want to provide an overview about the study and an update about where we are. So let's begin with an overview of the tominersen program. First of all, what is tominersen? So it's a non-selective Huntingtin-lowering antisense oligonucleotide that's delivered via intrathecal injection. And so it lowers both mutant Huntingtin and the wild-type Huntingtin, and we've been able to show conclusively across multiple studies that we can lower mutant Huntingtin in the cerebrospinal fluid of the CSF. Now, we believe that because mutant Huntingtin is a pathological driver of the disease, if we can lower mutant Huntingtin, then we hope that this will have an impact in terms of the progression of the disease. So tominersen is a really rich program history. It started with the Ionis-Roche partnership back in 2013 and led to what was really a breakthrough study, the phase 1/2A, where we could see that we could lower mutant Huntingtin in the CSFs of individuals with Huntington's disease. This then led, of course, to Generation HD1, which was the phase 3 study that I'll focus on today, and a number of other studies, open label extension, natural history, to name a few. Now, I think many people will know that back in March 2021, based on a planned review by the IDMC, a recommendation was made to stop dosing. And so Generation HD1 was halted. When this happened, we looked at the data in detail, and we were able to identify a subgroup that may have had potential benefit. They were younger with less advanced disease, with potential benefit, particularly at lower exposures, and this really formed the basis for Generation HD2, which I'll cover today. So here's an overview of the clinical development program. As I said, I'm going to start the talk and focus on Generation HD1. Generation HD1 had placebo and 2 treatment arms, 120 milligrams every 8 weeks, which we refer to as Q8, and 120 milligrams every 16 weeks, which we refer to as Q16. Importantly, Generation HD1 also had a loading dose. And the reason I highlight this is because I'm going to share some data from the GenExtend open label extension study where we didn't have a loading dose. And this is particularly relevant when we consider the NFL changes, which I'll come on to later in the talk. So what were the key findings in Generation HD1? The first thing that we can say is that there was clear evidence of target engagement. So we were able to lower CSF mutant huntingtin in a dose-dependent fashion. So here we're looking on the y-axis percentage change from baseline in CSF mutant huntingtin, and on the x-axis we're looking at the time point. And you can see if we focus on week 69, there's clear dose-dependent lowering. So placebo is in gray, orange is Q16, and blue is Q8. And the other thing to say is we achieved the predicted lowering that we estimated from our clinical PK/PD model. However, this didn't translate into the efficacy that we hoped for. So here we're looking at TFC, total functional capacity. On the y-axis, we're looking at change from baseline. Again, if we focus in on week 69, we can see that treatment every 16 weeks in orange was comparable to placebo. However, treatment every 8 weeks was worse than placebo. And this was the data that formed the decision by the IDMC and our subsequent decision to stop dosing on Generation HD1. So that was the efficacy data. Let's now look at the adverse event profiles. So the first thing I want to say is that at 120 milligrams every 16 weeks, the rate of serious adverse events and adverse events were comparable to placebo. This gives us confidence in the new study that with the lower doses, dosing every 16 weeks, we anticipate a similar adverse event profile. Of course, if we look at Q8, treatment every 8 weeks, there were more serious adverse events relative to placebo. So this is not a dosing regimen that we would look to go back to. Let's move on now to some of the biomarker changes that we saw. So here we're looking at ventricular volume. This is annualized ventricular volume. And the reason we annualize it is because ventricular volume increases as part of the natural history of the disease. So if you look at placebo here, you can see that there's a constant rate of ventricular volume increase. So the annualized ventricular volume is really the rate of increase that we're looking at here. So there's a constant rate in the natural history of the placebo group, but you see there's a dose-dependent increase in the rate on treatment. This goes up at week 37 and then begins to come back down at week 69 and comes down a little bit more after treatment was stopped when we look at week 101. And I'm going to come on to show some of the data to try and explain some of the thoughts around this. Here we're moving to CSF neurofilament light, or NFL. NFL is a measure of brain cell loss. And we can see here that there was an increase in NFL at week 21, and this was a significant increase in the treatment every 8-week group. We see an increase from baseline in the treatment every 16-week. However, it's still in line with placebo. The NFL increases then reduce at subsequent time points at weeks 37, and then at week 69, you can see that the confidence intervals all overlap with placebo. And again, I'm going to share some data around NFL in terms of the post-hoc analysis to try and understand this as well. So then let's move to the post-hoc analyses. First of all, when we looked at the data after dosing was stopped, we had a number of hypotheses that the efficacy may not have been what we had hoped for because people may have been too advanced in the disease. Individuals may have been older than anticipated, and the dosing may have been too high. So we identified a subgroup who were younger with less advanced disease. We referred to this group as the low age, low CAP group because it was based on a median split of age and the CAP score, the CAG, which is a function of CAG and age. Here we're looking at low age, low CAP, but we also split by exposure. So exposure is the level of tominersen in the CSF. Again, this is a median split. So green is low exposure, red is high exposure. The dotted line represents placebo. So you can see in this low age, low CAP, low exposure group, we see a potential benefit relative to placebo in TFC. We also see the similar patterns in cUHDRS and other elements of the Unified Huntington's Disease Rating Scale. And this is what's really formed the basis for Generation HD2 that I'll come on to talk about, where we're targeting low exposures. Let me share some of the post hoc analyses where we try to understand the implications of the ventricular volume. One of the key questions that we had was whether the increases in ventricular volume had any impact in terms of clinical outcome. So in this plot, you can see there's a number of different rows where we're looking at the different elements of the UHDRS. So we've got composite UHDRS, SDMT and strip word reading, which are cognitive measures, TFC, which is the functional measure, and TMS, which is the motor measure. And we're looking at placebo, Q16, and Q8. And on the y-axis, we're looking at the change in clinical endpoint up to week 69. On the x-axis, we're looking at the change in ventricular volume. And the take-home message from this is that there are no linear correlations. So essentially, people that have increases in ventricular volume don't have worse outcomes, at least in a linear fashion. The next thing we wanted to try and understand was whether increases in ventricular volume may reflect atrophy. And I think this was, of course, an understandable concern because this is typically what happens in natural history. However, when we look at this in tominersen, we don't see increases in ventricular volume in the same groups. We don't see corresponding losses of whole brain volume. So it suggests that it's not related to atrophy and it's a different process that's going on. We also have shared data before where we don't see any linear correlations with increases in NFL and increases in ventricular volume, again suggesting that it's not atrophy but some other process. Let me now share some additional data on NFL or neurofilament light. So as I mentioned, in the GenExtend study, we had individuals that didn't have a loading dose. And this is because they either came from the natural history study that we had, or they came from the placebo group of Generation HD1. So they didn't have a loading dose in contrast to those that were on Generation HD1 that did have a loading dose. So we can look at the treatment every 16 weeks at 120 milligrams and compare load with no load with Gen HD1. And what we see is that those that didn't have a loading dose don't have increases in NFL above baseline. So in the new study in Generation HD2, we don't have a loading dose because we hope that we can avoid the increases in NFL, particularly at the lower doses, but also without the loading dose. Here we're looking at plasma NFL. And the dotted line here represents placebo. Again, Q8 is in blue, Q16 is in orange. So we see the NFL increases again at week 21. But the important piece of information here is that for Q16, 120 milligrams, subsequent to week 21, we see NFL go down relative to placebo. So this gives us hope that in the new study with the lower doses and the no loading dose, we can avoid an NFL increase. And we hope that then this will allow us to see NFL lowering to a greater extent and potentially earlier than we see it here. Let's move on now to exposure response analysis. And these analyses can give us an idea of what happens when we dial down the dose. So just let me explain what we're looking at. So here we pulled the data across Q8 and Q16, 120 milligrams. And then we can look at the level of tominersen in the CSF and split individuals into quartiles. So we can look at highest exposures and lowest exposures and compare this to placebo. So yellow here is ventricular volume. This isn't annualized. Pink is CSF mutant Huntington and red is NFL. So you can see at the highest exposure, you have NFL increases, you have an increase in ventricular volume, and you have CSF mutant Huntington lowering of around 50%. But at the lowest exposure quartile, this is the exposures that we're targeting in Generation HD2. You avoid that NFL increase. The increase in ventricular volume is just slightly more than placebo, but you still have CSF mutant Huntington lowering of around 25 to 30%. So this gives us confidence that we can avoid the unfavorable biomarker changes that we've seen in Generation HD1. Now moving on to some of the digital data. So we had digital measures across many of our Huntington's disease studies. And so we used this to identify the most sensitive measures in terms of monitoring progression in Huntington's disease. We developed this using the open label extension on natural history studies and also the observational digital HD study. And then we validated this using Generation HD1. And then, as many of you may have heard, we had differential dropout with the digital measures. So different people in different treatment arms and different numbers in treatment arms and placebo stopped doing the digital measures. So we've applied a statistical technique called propensity score weighting to address the different groups had different baselines. And so then we can make inferences about what happened on the study. Here we're looking at those that completed digital data up till week 52 and up to week 68 to just ensure consistency. And we can see here that if we look at the last time points, there's a trend towards potential benefit for Q16 relative to placebo and Q8. So you can see this for those that completed the measures up to week 52, but also up to week 68. Now, it's important to remember that those that completed the digital data had less advanced disease than those that stopped doing the digital measures. And so this pattern of potential benefit is consistent with our low age, low cap analysis, where individuals who have a less advanced disease seem to do better on tominersen. So let me now move to the question about how much tominersen gets into the brain and what brain regions. So this is data from Blair that he's presented back at CHDI last year. And here we're looking at a single post-mortem case where the last dose was 72 days from the last dose. In the plot here, we can look at the tissue concentration of tominersen across different brain regions. And so, as expected, you get less tominersen into the deep brain structures, particularly the caudate, but I think it's important to say you do get tominersen into the caudate. You also get it in the thalamus and across the cortical regions. And this is really consistent with our comprehensive preclinical data from non-human primates. And I haven't had time to share it today, but the Huntington mRNA data is very much consistent with this, where we see lowering to a similar extent in these brain regions. So now I just want to switch gears and give an overview of Generation HD2. So I've already covered a lot of this data, but I just want to summarize it. And this is what gives us reasons for optimism for Generation HD2. So we know we can get tominersen into the selectively vulnerable brain regions in HD, cortex, striatum, and thalamus. We've conclusively shown across multiple studies that we can lower CSF mutant Huntington. The safety or the adverse event profile in Q16 was comparable to placebo in Generation HD1. Therefore, we anticipate similar when we're looking at the lower doses in Generation HD2. In terms of clinical outcomes, we've seen with the low age, low cap, low exposure group, this potential benefit that is also supported by the HD digital motor score. And in terms of biomarkers, at the exposures, the lower exposures we're targeting, we believe that we can avoid the NFL increases, we can mitigate the changes in ventricular volume, and we also have potential for NFL lowering. So let me just go through the rationale for Generation HD2 and testing a refined hypothesis. So based on the post hoc analysis data that I've shared, we're focusing on these younger individuals with less advanced disease. So we're recruiting prodromal and early manifest individuals with HD. We're looking at lower and less frequent dosing, so 100 milligrams and 60 milligrams given every 16 weeks with no loading dose. And we're focusing on safety, biomarkers, and looking for trends of clinical efficacy. So just an overview of the study design. So we have 100 individuals per arm, roughly 17 prodromal and 83 manifest. Individuals will go through a screening process and they'll be randomized to 100 milligrams or 60 milligrams every 16 weeks relative to placebo. Participants will complete 16 months blinded treatment and then continue on until the last participant has completed their last visit. This is what's known as a common close. We'll have a focus on safety, so the IDMC will monitor clinical, biomarker, and safety data every 4 to 6 months. And if the data supports it, we'll then move on to an optional open label extension study. So what are the key differences in Generation HD2 compared to Generation HD1? As I mentioned, we don't have a loading dose. So you can see here on the top, there was the dose given at randomization and then a loading dose given 4 weeks after. We don't have this in Generation HD2. Also, the visits are much less frequent because we're only dosing every 16 weeks. And we have a 9-month visit where we sample between dosing to understand what the maximum Huntington lowering is that we can achieve. And as I mentioned, we're looking at lower doses, 100 milligrams or 60 milligrams versus the 120. And as I said, reduced dosing frequency, so we won't be going back to the treatment every 8 weeks. There's no loading dose, and as I mentioned, the CSF sampling. So I'm really excited to say that we're now more than 50% recruited. So the study is well underway. We're active in 15 countries and 70 sites. And as of September last year, the IDMC have done their first review of the data, and they've recommended to continue as planned. So the study is well underway. And with that, I'd like to thank you for taking the time to listen, and I'm looking forward to the discussion and any questions.
Yeah, thank you very much, Peter. So we are starting with our discussion part.
Yes, so thank you very much, Peter, for this information. Every time you speak, there are new information, and every time you speak, I understand better what is all going on. This is just great. And we do, as a community, appreciate very much that Roche, that yourself are so open to discuss all the detail of the study. And from the publication, which just came out now, we have also quite a large number of additional data, which is very interesting and which gives rise to quite some discussion. So let's start with a couple of questions. As a chair, you always have the opportunity to ask your own questions and get a better understanding. But then we, of course, are going to have a larger discussion. I am quite interested in the biomarker part of it, and I would like to get some comments on the neurofilament light change. So I jump already directly to details, but of course we can have overarching discussions afterwards, as it were. Now, maybe Ed, you have worked a lot on NFL. Would you mind to comment a little bit on the course of the NFL and also the differences we see in NFL and additional clinical data, the comparison between week 21 and week 69. Ed, if you would mind to say some comments on that, maybe to get started.
Yeah, sure. Thanks, Jean-Marc. And thanks, Peter. Hi, everyone. So neurofilament light, for anyone who doesn't know, is a protein that's a structural component of axons. So more or less, whenever neurofilament goes up in spinal fluid or blood, that is a sign that the rate of damage or death to neurons or axons has increased. And that's usually bad. When it goes down, that's usually a sign that some form of damage has been slowed or prevented, and that's usually been good. And we've known for a while that treatment with high milligram doses of tominersen was associated with these unexpected short-term increases in NFL, in CSF. That was seen in the phase 1b/2a, excuse me, first in human study that started in 2015. Of course, no one had ever lowered Huntington in human nervous system before. And so there was a lot of wondering about what that might mean at the time. And it was also neurofilament, although it was relatively well established in other conditions such as Alzheimer's and motor neuron disease and multiple sclerosis and traumatic brain injury, it hadn't been studied extensively in Huntington's disease. And so this, I think, to me, one of the major positives that has come from this obviously highly undesirable outcome from Generation HD1 was what we've learned about the biomarkers in general and neurofilament light in particular. And so increasingly, I am inclined to think about NFL as a biomarker of the therapeutic window in Huntington's disease. It remains to be seen whether it will meet that conception that I have of it. But what I mean by that is the therapeutic window is where we want to land. And with every medication, more or less, if you underdose, you won't achieve the desired effect. If you overdose, you will achieve undesirable effects. And the therapeutic window is the sweet spot in between those two extremes. Medications can have a broad therapeutic window or a narrow therapeutic window. And I think what we seem to be uncovering for neurofilament is that when it goes up in Huntington's disease, it means the same, broadly speaking, so far, as what it has meant in other conditions. In other words, that neurons are being damaged. When it comes down, we've reason to expect that that should be associated with benefit. So I think it's, for me, one thing that's really encouraging is one of the things that Peter said about neurofilament in the so-called natural history study, which was where patients were followed for a while and were then rolled over into open label treatment with the drug. But that was the first time that anyone had been started on tominersen without a loading dose. And we already know from that experiment that you can give doses of tominersen that are expected to lower Huntington significantly, that don't produce a transient increase in neurofilament. And that's even at 120 milligrams. So, I think neurofilament seems to be emerging as a biomarker that can tell us about safety or perhaps future expectation of adverse outcomes. I think it's really encouraging that there seems to be a suggestion in the 16-week that the neurofilament in blood was below baseline by the end of the study. That is, a subgroup analysis, unplanned, and needs to be replicated. But I think we have reason to expect that neurofilament will deliver, and I think it's telling that neurofilament now is widely used across therapeutic trials in Huntington's disease, and at least behind the scenes, drug companies are paying close attention to whether neurofilament produces short-term increases and whether it seems to be producing the kind of downward trends we'd like to see. I'm sorry, Jean-Marc, you had a second question, but I can't remember what it was.
Oh, I have many questions to many people. Thank you very much, Ed. Related to a similar question is the fact about the data about protein and leukocytes. By the way, the NFL did not very much correlate with any of the clinical markers at week 69 and 21, which is still something which can also be debated at some point of time. But now my question here is about the leukocyte and protein. So as neurologists, we in general, we don't like so much of protein elevation, neither of leukocyte and elevation when we perform a lumbar puncture. Bernard, would you like to comment on this question? And also a related question to the one I asked to Ed is about this difference between week 69, week 21, and in relation with the clinical aspects, markers and the measurements. Bernard.
Sure, pleasure. What we would like to understand really is why we had a biomarker of target engagement going in the right direction and no clinical signs of improvement despite that. And one of the potential explanations for this is that there are side effects induced by tominersen, potentially dose-dependent side effects. And in that context, it is that I believe we should discuss the indication for an increase in leukocyte count and an increase in protein content. And this is something that has been observed with other antisense oligonucleotides as well. And I believe it's related to, or I favor the view that this is related, this indication of a change in CSF flow and potentially for inflammatory reactions induced by the antisense oligonucleotides, and that may be related to the dose-dependent increase in size of ventricular volume, despite the fact that parenchyma stays the same. So I favor to interpret these changes in CSF protein content as an indication of changing CSF dynamics, in line with the concept of a disbalance between production of CSF and reabsorption of CSF, resulting in enlargement of ventricles. And despite the fact that there is no change in brain parenchyma, and I believe there's data showing that, as you would expect, the size of the CSF space over the convexities is indeed contracting, consistent with this overall concept. So I think it's a potential illustration for a dose-dependent toxic effect of ASO.
Thanks. And the context here is, or the discussion is between, as you mentioned, circulation, CSF circulation by itself, which, if it is changed, may by itself also raise the protein levels in the CSF, and the actual inflammation. And Blair, your data about public autopsy case has been briefly mentioned by Peter. Would you like to comment on those data, and also whether these data help us in sorting out these two different possible pathomechanisms in terms of either CSF circulation and inflammation, or both? Probably is both, but Blair, please, if you want to comment on that.
So I would like to caution overinterpretation of a single assessment of one postmortem brain. I think I don't believe our analysis is providing any real insight into the cause of the ventricular volume increases. I think what the postmortem data is most valuable for is our ability to assess for the first time the levels of tominersen in tissue following dosing, and then subsequently the amount of Huntington mRNA lowering that we see in the various brain regions. And despite the fact that it was an extended period, 78 days, I believe, after the last dose, there was still a very significant lowering of mRNA compared to the best matched controls we could provide in a variety of brain regions, including deep brain structures such as the thalamus, and also significant lowering in hippocampus, as well as caudate and other cortical regions. So I think what that tells us is that tominersen is a very potent drug. It is hitting its target. It is penetrating even into deep brain structures, and it is lowering, having an effect on total Huntington lowering. I think when we start to think about the CSF volume space differences, and specifically the ventricle, again, we can look at CSF spaces in various regions, and as Bernhardt suggested, if it's an ex vacuo phenomena, you should see increased CSF spaces everywhere, and you should be able to see changes in the parenchyma, in the brain tissue itself. In the absence of that, and in the context that it may be reversible, and that to me does not suggest that it's an ex vacuo phenomena. The fact that the cortical CSF spaces are actually contracted and the ventricular spaces are larger. I think there will be emerging data that other Huntington lowering approaches may also have effect on ventricular volume. And I think suggesting if it is reversible, that it's more likely something to do with the CSF flow rather than a loss of brain tissue. So I don't know that my case or the postmortem case that we've looked at provides direct insight into the ventricular volume increases. I think Roche has done a very good job of looking at any correlates with those changes in ventricular volume. And my personal feeling is it's probably we can't rule out a class effect of ASO toxicity, but it may very well be a wild type Huntington lowering effect and may have something to do with the total Huntington lowering that we're seeing with this particular ASO.
Yes, and Anne was providing exactly a comment on that aspect, whether the loss of Huntington function may play a role in the CSF circulation. And you like to comment further on that?
Yes, I was just going to pick up on what you said, Blair. Yeah, I think it's worth remembering that although it's very likely and very possible that there are toxicities here that are related to the ASO, it's important not to lose sight of the fact that we've lowered wild type Huntington here, and what part that's playing in this. We can't untangle that in any of these studies. I don't know whether that's something that Fred would like to comment on as well.
Yeah, absolutely. So thanks, Anna. So indeed, I think it is incredible the effect of tominersen in the target engagement in its capacity to reduce HTT both alleles. And indeed, we have published a couple of years ago the role of the silencing of reducing HTT wild type levels that has a strong effect in CSF circulation and on the size of the cilia that are within the ventricles. So we could show that actually reducing HTT in wild type animals leads to a defect in CSF circulation. So therefore, I mean, this could account for the decrease in the size of the ventricle, which actually is proportional to the dose, and that's quite amazing. So maybe here what we do see is really affecting CSF circulation. So that could fit, and which could be different from the rise in NFL that occurs at the beginning. So that would be my interpretation of the results regarding the increase of the ventricle.
Thank you, Frederic, for these comments. Maybe another comment directed to you also as a scientist working also on basic questions. It does what we see here lead us to discuss about the approach, whether it's a right approach to go through lowering MTT lowering, or is it for you the question still open, or would you provide some more general and basic comments on that question, please?
Well, I would tend to say that, of course, I mean, we would certainly prefer to have like a selective silencing to get rid of the mutant HTT and keep the wild type copy. The problem we may face, actually, even with the lowering of the specific mutant allele, is the fact that we would be with only one copy left as compared to the wild type situation. And this situation would be within patients that for many decades, they have been carrying both the wild type and the mutant copy. So they have already, and that's also the work from the group of Sandrina was shown that there are already developmental defects occurring very early on that the organism can cope with. But you can think that even one copy might not be sufficient to one wild type copy might not be sufficient to rescue the deficit. So this is still a very open question, I'd say.
Thanks for this comment. to get a step further, or to come back to some aspects of our discussion so far, we have the lowering of HTT. We have, however, a decrease or not an improvement in clinical parts. Could that be? It's a difficult question, I guess, but what is the balance between the side effect and the appropriate effect? Can we separate these from the data we have at hand? Maybe Sarah, you would like to comment on that question, if I have made it clearly, the balance between side effect and the potential clinical effect. Can we sort this out, dissect from those data?
I think that's a really important question, Jean-Marc, and I don't think the Generation HD1 data is going to allow us to answer that. I think I agree with Bernard. I think there's evidence of ASO backbone induced inflammation. I absolutely agree about wild-type huntingtin lowering and the ventricular enlargement. And I think there is beginning to be a body of data suggesting that wild-type huntingtin lowering is playing a role in the ventricular enlargement based on data from other studies, potentially, and ongoing clinical trials with other agents are going to help us dissect this. But it is becoming a theme of ventricular enlargement in total huntingtin lowering studies. But we can't dissect that in Generation HD1 from potential ASO toxicity. In terms of benefit, I think the post-hoc analysis is a post-hoc analysis which generates a new hypothesis, which is tested in Generation HD2. The question everyone often asks is, if the 16-week group had been taken to the full 2 years, would there have potentially been some benefit clinically in the 16-week group? That's a really important question that we are never going to have the answer for, because they decided to close down both arms. And the data from NFL and the digital motor scores is interesting in that respect, in that there may potentially have been a readout. I also think there is an important question here for the field, and everything we learn is going to be iterative, i.e. a step at a time, but it may be that we do need an allele selective approach. We know that we have to target the CAG repeat expansion as well. And that looks like it's the rate driver of disease. And so that's another important area that's not targeted by a total huntingtin lowering therapy. And this tominersen doesn't target exon 1. So we don't yet know. In mice, I think it's pretty clear that exon 1 plays a role in the pathogenesis. I think in mice, I think I'm very supportive of that mechanism. We don't yet know in humans, and there are approaches that are targeting exon 1 in development and also in trials. But again, there's the link between lowering total huntingtin that may muddy that slightly. So I think everything that we're doing in trials it will continue to learn. And I think the data from Generation HD2 will be important in telling us if there's a lower dose that's safe, that has a good therapeutic index, as Ed was saying. Do we see anything with NFL? I would hope that we would mitigate the problems with the ventricles, but let's see if we see an effect on clinical measures in time. So it's a long answer, but really to say there are lots of different issues that Generation HD1 gave rise to that we can't disentangle with that data. But as we do all the different studies that are ongoing, I think we will get more data in an iterative way that helps us in our goals to try and find an effective disease-modifying therapy.
Thanks, Sarah. Speaking about clinical data, we have seen very briefly the data from the digital measurements, But still, the major clinical presentation or the data are used from the clinical presentation. So I would like to ask maybe Patrick, since you have been working on that yourself, what are your comments or what is your take about this difference of so much of digital data being sampled and actually being less prominent in the present analysis? Would you like to comment, Patrick?
Yeah, sure. Thank you. First of all, I want to say also it was very interesting to follow this new presentation with the new aspects. I think the digital readouts I have less high expectations about because we're very early still in the development of this information intervention. I think if you don't see a lot here, that's because the intervention doesn't do enough good at the moment. I think it's important to, at the moment, focus on biomarkers and safety aspects. And for the biomarkers, I'm just happy that they're being done, and this is like a feasibility or a rehearsal of that we're getting ready to capture digitally what will hopefully be clinical benefits in ongoing and future trials.
Thanks, Patrick. I would like to come back to Peter, of course. Thanks again for your presentation, but there are still questions. Would you like to have a general comment or a specific comment to what has been said so far?
I agree around the question of the ventricular volume. I think it's difficult to disentangle whether this may be related to wild type or the ASO specifically. I think that the data from generation HD1 can't disentangle those two things because the Huntington lowering, the non-selective lowering is dose dependent. To Bernard's comments, I think that at the higher exposures we see at NFL go up, we see increases in white cells and proteins. When we dial that down to the exposures that we're now targeting, we can avoid that. So I think that there is signs of inflammation at the highest exposures, but our focus in this study is to avoid that with the lower doses that we're using.
Thanks. And a precise question. Now we can come to the question which has been put into the Q&A part of it. Some will be answered later. We understand that. But I have a question here about do we have enough safety data and a good enough safety monitoring in place to justify additional research trial now with tominersen? This is a critical question, but probably an important one. What is your comment, Peter, on that?
The first point I would make is that when we look at the safety data from generation HD1, the adverse event profile for Q16, 120 milligrams, was comparable to placebo. Now, we're looking at lower doses relative to that. We're not having a loading dose, so we would anticipate a similar adverse event profile to be comparable with placebo in the new study. Of course, safety is one of our key focus areas in the new study, so we have an independent data monitoring committee, an IDMC, to look at clinical imaging and fluid biomarker data every 4 to 6 months. So I think while we anticipate an adverse event profile similar to placebo, we have the checks and balances in place to monitor that at regular intervals.
Thanks. Another question is whether tominersen was still in the brain after 72 days. This is we don't have data directly from the brain, but can we relate this from the data, the CSF data?
It's a great question.
Anybody else would like to comment on that? Peter, go ahead.
I think I agree with you. It's difficult to be able to measure half-life in the brain, but we can look at our predictions from the clinical PK/PD model, and based on those, we would expect Huntington to return back to normal levels after around 9 months.
And a related question, also from Hervé Lange, is about what happens after a longer term in ventricular volume after 1 and 2 years. Are they still present, or is it also recovering?
So
the ventricular volumes.
yeah, so from generation HD1, we have data going up to week 101. What we can see here is that the rate of increases, even while on drug, but also after drug was stopped, the rate of increases begin to decline back towards normal levels. I think that one thing I would say about the ventricular volume measurement is it's an exploratory biomarker, so it's what's known as boundary shift integral, and I think this is distinct from when we see clinical hydrocephalus. The reason I would make a distinction between these two measures is because with VBSI, it's unclear what the clinical relevance of this is. When I showed the data looking at change in VBSI and change in clinical endpoints, we don't see a relationship. So I think it's still difficult to understand what this means. And I mean, to Bernard's points, that this may be due to changes in CSF flow and changes in the CSF spaces that we see in the sulci relative to the ventricles. So I think there's some unknowns we still have with this.
Thanks. Another question is about, again, about CSF MHTT lowering, which is variable among trial participants in each of those of the dose cohorts. And then by targeting 25% lowering, is there more variability? Who would like to comment? I saw no hand raised, so please.
I think only Peter can answer that.
I'm happy to answer that. So when I'm talking about the 20 to 25% that we target in generation HD2, those are based on medians from the simulations that we run in a typical low age population. So there's variability around CSF mutant Huntington. I think the variability with more lowering and with less lowering is comparable. So I don't think that there's more variability with less lowering that we're doing in the new study.
Thanks. Another specific question was what was included in HD-DMS?
The HD digital motor score. So we have speeded tapping, chorea, gait, and turning, I think, from memory.
Balance, Peter.
Balance. Thanks. Thanks for reminding me.
It's can I comment briefly, just because I've been involved in the development of that, the DMS, which will be presented in a couple of weeks at the Therapeutics Conference. So the Roche digital biomarker platform consisted of a smartphone and watch, which were used to collect passive data all the time. The participants were wearing them, and then from time to time, rather more frequently than we would do it now, they were asked to participate in active motor tasks, which were all based on the smartphone. And obviously, as with any digital measurement platform, thousands of different parameters are collected every time these tests are performed, and so there's a huge risk of multiple comparisons. And you know what? The one thing we don't want to do is cherry pick the data to provide evidence of benefit, or indeed evidence that the digital motor platform might be better than it actually is. So a really stringent set of non-hypothesis driven criteria were used to winnow down the digital parameters that were eventually then put to the test in the digital motor score by the Roche digital biomarker team. And the four that sort of rose to the surface as in terms of having the best signal to noise ratio and the best ability to predict future progression in Huntington's disease were these four: the balance feature, which actually measures truncal chorea, the measure of chorea in the upper limbs, tapping, and the variability of step power. And so that's now a kind of predefined set of characteristics that will then hopefully be used to produce a much more focused digital motor assessment for future natural history and clinical trials.
Thanks. I have another question. Actually, it's a question of mine. In the paper, there is an additional figure about the dropout probability, and it's interesting to see that age are fewer dropouts, being male also fewer dropouts, and so on. Would someone like to comment on this question of dropout and what kind of conclusions can we say? This is a figure 1 of the supplementary material. I was surprised that CUHDRS at baseline is related to having more dropouts also, for example. Peter.
Yeah, so I think for me, the main takeaway on this is that the worse your CUHDRS is at baseline, the more likely you were to drop out. It may have been that individuals with more advanced disease had more difficulty performing the tasks. But I think that for me, when we look at the analysis that I shared, we're really looking at individuals with less advanced disease. So that potential benefit that you see in Q16 in the digital analysis, it's a very different analysis, but it fits with the low age, low cap post-hoc analysis as well. This idea that people who have less advanced disease have the potential to benefit from tominersen. So I think these analyses, they confirm what our initial hypothesis was.
Great. It's great to see that higher age is more related to steadiness in what you do. This is reconforting. Now, any further questions from the panel to each one another or to Peter? And I see your hand. Yeah.
Could I ask Peter very quickly, because I think it's interesting that you can look at the dose and you can look at the exposure, and sometimes you get different answers depending on how you do that. What have you found to be predictors of higher exposure? In other words, what are the properties of a human being that causes 120 milligram dose to produce a higher concentration of drug in the CSF? And is that something that we can take forward when it comes to designing future ASO trials to sort of customize perhaps?
So I think things like BMI can have an impact. I think it's difficult to take these things forward given the variability within the variability between people to be able to go to personalized medicine, for example. But I think the exposure analyses can inform us in terms of if we were on the right ballpark in terms of dose and maybe looking at lower doses.
Anne, you would like to say something because I see you have just unmuted yourself.
Oh, yes. No, I just had I was very interested in the post-mortem study, and of course it gives us a lot of reassurance that the primate models are useful, which is nice. But I wondered what the conclusions were in terms of the route of delivery, because it was striking to me how low the levels of tominersen appeared to be in the caudate, and that there didn't appear to be any putamen measurements, or did I miss that?
There were no. So I assume I'm going to answer that. There were no putamen analyses done as of yet, and overall, the predictive models did have a reasonable correlation with what was observed in this N of 1. I would say that overall, the amount, and to answer a previous question, there absolutely was tominersen that is measured in the brain, even 72 days after the last dose. So there's no question. That's absolutely clear. That's one thing that this data tells us. And the levels of Huntington lowering, as best we can estimate them, were in line with the predictions, but in almost every case, slightly greater than would be predicted. So overall, not to overinterpret it, I think that we have to be very careful about extrapolating from an indirect measure such as CSF mutant huntingtin levels, which are taken at trough. So those levels are just prior to the next dose, and extrapolating with certainty. I think overall the predictive data was good, but the amount of huntingtin lowering and the amount of tominersen seen overall was more than predicted. So in thalamus and hippocampus, the huntingtin lowering looked actually quite a bit more impressive than one would have predicted, and those are deep gray structures. What this tells me overall is the predictive models have value and were accurate. This is a very potent huntingtin lowering drug. And much like chemotherapy, when you use a very potent drug, you have to learn to use it properly, and there's going to be risks and benefits. And we have one trial, one properly done, well-powered, double-blind, placebo-controlled trial on which to base future data. All we can do from that study is learn from it, design better studies, and learn better about how to use this drug. There were many, many, many critics when chemotherapy first came out saying this is poison, it will never work. Cancer therapy has dramatically improved through iterative scientific advancements. We need to do the same thing. And in fact, Roche is, to their credit, doing this very carefully and moving forward and advancing with appropriate next step studies. I think it's not a surprise. This drug was chosen because it was incredibly potent at lowering huntingtin. And I think the data all suggest it is very potent at that. What we need to do now is understand, is there a window of opportunity, a therapeutic window where we can get benefit while minimizing risk and potential side effects?
Thanks for these comments, which almost sound like concluding comments exactly at the right time. Any further comments from the panel? Doesn't seem to be the case. So I would like again to thank Roche, to thank especially Peter McCollum for his presentation, which has given rise to such a lively discussion. Thanks to all the discussants, and of course, thanks to Yuri as well, who has prepared the technical part of it, and not only the technical, but also has written some questions which we could discuss. And thank also to all the participants, and specifically the participants who have asked questions. Some of those questions will be answered in a later period of time, or has been answered implicitly and will be more explicitly by someone on directly, in a direct way. So, Yuri, anything else we should say?
Thank you very much, Jean-Marc, for this conclusion of the meeting. I think we're done perfectly in time. And like you mentioned, we will post the answers to the questions that were not answered live later on with the recording of the meeting. Thank you very much again, and have a nice rest of the day.